Epigenetics using CRISPR-Cas9. Recently, in the Salk Institute in the Belmont Labs, they have been investigating ways to alter the activity of genes without changing the underlying sequence. The lab has found a technique using a modified Cas9 system that does not cut DNA. This is helpful because using CRISPR-Cas9 to cut DNA can lead to harmful mutations to be introduced. This new system, created by the Belmont Labs, can alter the phenotype of an animal with an epigenetic editing technology, preserving its DNA from mistakes. Let's be clear, this is not gene editing, although it involves a gene editing tool. To create this technique, first the lab inserted the gene for the Cas9 enzyme into one AAV virus. Along with the enzyme, they inserted a short, single guide RNA, sgRNA, which specifies the precise location in the genome where Cas9 will bind, like a GPS. And lastly, a transcriptional activator, kind of like a gene switch. The shorter sgRNA, which is only 14 or 15 nucleotides compared with the standard 20 nucleotides, which is used in most CRISPR-Cas9 techniques, prevents Cas9 from cutting the DNA. However, all of that was too big to fit in one AAV virus, so the lab split the components into two viruses. One had the Cas9 enzyme, and the other had the GPS system and the gene switches. In theory, the two viruses would supposedly work together after both being injected into a patient. Preliminary experiments suggest that it is safe and doesn't produce any gene mutations. In a mouse model with acute kidney disease, the technique activated previously damaged or repressed genes to restore normal kidney function. They were also able to induce some liver cells to become pancreatic cells, which produce insulin. This can be used to get rid of type 1 diabetes. This could possibly eventually be used in neurological diseases such as Alzheimer's or Parkinson's disease. Overall, this new technique, using epigenetics and a modified Cas9 system, has a bright future because it doesn't cause mutations and all mouse testing has been fairly successful. However, with this new technique comes some ethical issues and remaining challenges. Ethically speaking, we have to consider how we are to use this amazing editing tool. Who can use it? How can they use it? Also, can we just use it to edit babies or people or their organs? Should we let natural selection take its course? And what if we use it to edit a species, then they accidentally escape and wreak havoc on the natural ecosystem? Regulation is truly the biggest challenge that is left to deal with. We must answer these questions with strict policies that don't leave any room for these tools to fall into the hands of careless people or scientists with bad intentions.